sorry.
Okay.
Yeah.
my goodness, thank you. That's kind of introduction my mother would have loved to have heard. So I'm Kevin Moore, it says so right here. And I'm going to, in the next, it won't be 2 hours, but the next hour and a half to hour and 40 minutes, give you an idea, an overview, okay, let's do it that way, of large molecule drug development. And for the advisors who have been at, I've given this talk 3 other times. It's not hugely different. So you can take a nap or talk amongst yourselves or think of even more discerning questions to ask than you did last time. For the students and fellows who are new and haven't seen it before, I have added some additional material since I have the whole session to myself this time. Oops. Okay, so I won't go through this. Kevin covered most of this. The visiting distinguished fellow chemocentrics, that was my first exposure to the small molecule world. And it was a really nice place to be, really liked the people, but it really taught me that small molecules are not my thing. I don't think well about them, and I went back to biologics. But especially it was much better than being an extinguished fellow. So anyway, why am I talking about this topic? So I've been around. I worked in discovery research, and for much of the stuff I worked on at Amgen, it's still, they aren't disclosing it publicly, so I can't really talk about that. And importantly, it's the kind of stuff you talk about when you get old, like I am. So, gosh, did it freeze so quickly? This must not like Kevin's. It did it to, oh, there we are, okay. So, some disclaimers, any opinions that I express are mine. They don't represent Amgen or any other previous employer, whether it exists or not. And in fact, 3 of the 5 places I worked do not exist any longer. They were gobbled up by acquisition. Schering-Plough acquired DNAX very early on, and that was actually a good thing. That enabled us to do all the really cool science that was done at DNAX. After I left, they were acquired by Merck, of course at Chemocentrics, and then Avidia was acquired by Amgen. So I want to digress from the main topic for a few minutes for the, I hope, the benefit of the fellows and the students. So I was fortunate to have experience in large pharma companies, a very large biotech, 2 startups, and 1 not really startup, but a privately held, relatively small biotechnology company, even though it was small molecule focused. So I got to experience a lot of different things. And for those of you, so I'm going to offer the following, I hope. Let me try this. Okay. So I'm going to compare sort of larger biotechnology, and pharma, and the good and bad things of both, and I hope some of the students will benefit from this. So for the larger biotechnology and pharma, coming from my experience at Schering-Plough and Amgen and others whom I've talked to in other companies. So what's the good aspect of working in a situation like that? Well, there are a number of them. You have considerable resources. You can accomplish a lot by leveraging those resources. It's really quite remarkable. You can tackle longer term, more challenging and more important problems because you have those resources and because you're not, trying to scramble to, necessarily scramble to generate results for the next VC meeting, on Wednesday. There's a real emphasis on quality, and because substantial amounts of money and resources are going to be invested if the project you're working on becomes a development candidate or a drug. So the science really has to be the best that you can deliver. Sometimes to the point of obsessive compulsive disorder, I must say. Emphasis more, sometimes I'll have to say more on process than on output. But by and large, you can do really good quality work in that environment. there's the time and resources and mentoring that's really important, available to become truly excellent at what you do. And they offer stability. Now, these days, whether it's in healthcare-related fields or technology or whatever, there really isn't any such thing as a secure long-term position. That's just the facts of life. But the larger companies do offer stability, relatively speaking, compared to smaller companies. And it's rare to bet the company on a single product or technology. And you have a better opportunity for work-life balance. You know, if you decide to do something really unusual, like have a family, you know, this might be a more congenial environment in which to do that. So what's, come on. Sorry. Oh, okay. I got it. So let's see what happens. So like anything else, there's a bright side and a dark side. So large companies tend to have a lot of bureaucracy, may not be very nimble, and decision-making may be slow. The business-related decisions and portfolio considerations can sometimes overrule the actual science in terms of decision-making as far as what projects will be moved forward in development and which will be put on the shelf. And some companies, particularly the largest ones, are often unwilling to give a new therapeutic area group the time to mature and become productive. New management, just like, you know, when someone new comes into something, they want to shake things up and change stuff and put their own ideas and people in place. New management always needs to make their mark, and that can be a good thing or it can be bad. I refer to it as Pfizer disease. Those of you who've been in Pfizer probably know what I'm talking about. And we don't build medicines, we build careers. So that can be a dark side of working in a larger company, but it varies from company to company. There are varying attitudes towards and patience for innovation. That's just a fact of life. And there's a potential for people to become to find themselves in their own little pigeonhole. They'll do the same thing year after year, and it's not necessarily, do you have, you know, is it clear to you and the people who are working with you what your real impact is? And there's a comparatively, on the financial side, there's a comparatively smaller chance for equity appreciation. So I can see what it doesn't. It doesn't like to change the slide. That's what it doesn't like to do. All right. So what about startups? So the bright side, there's a potential to really innovate and maybe change the game of how medicine is practiced. Big motivator it was for me. High energy work environment, timelines are very aggressive. But everyone, your colleagues and you are pulling in the same direction can be very invigorating. Your work has a clear impact. Whatever you're doing in a small company, you are needed, you are critical. And if you fall down on your part of it, it's going to have a real impact on how things move forward for the company as a whole. You get to wear multiple hats and learn new things. You have to learn them on the fly, and sometimes you don't have the time to learn them deeply. You sort of learn them well enough, sort of in a fake it until you make it type of way. There can be equity appreciation if you're acquired or there's an IPO, but keep in mind that that's not automatic. It's only if the CEO and the financial officer, the VC investors, and the board of directors decide to be generous. I think we really should change to this if we can. So, all right, I'll talk of the slide while we're changing. But the dark side, limited resources, investor impatience versus long development timelines in this type of industry. Many small companies can be like I say here, a one-trick pony and live or die with a single technology or product candidate. There can be, because of few resources and small numbers of employees, there can be corner cutting and many temptations to shade the truth, particularly to investors. Okay, we're going to, we switch over. All right, sorry, I lost my timing there. Okay, but I don't see it here. Anyway, if I'm not seeing it here, I can just keep looking up there, I guess. Okay, we're almost there, great. Okay, thank you, son. Sorry about that. All right. So we went over limited resources, corner cutting, temptation to shade their desperation when there's not enough, when there's too much month left at the end of money. That's the definition of desperation. And the honest truth is many, if not most, small companies eventually circle the drain and go down. Things don't work out. That's just the nature of our business. This should work. Oh, we're back to where we were. Okay. Right, so my point in all this is, I hope it gives the students and postdocs an idea of what the pluses and minuses of these career paths are. I'm very grateful to have been able to sample all of them. I think in retrospect, the lesson I learned that the times in your life to take the big risks in a small company are either at the very beginning of your career or at the very end. And in the middle, maybe from your mid-30s through your early 50s is the time maybe to have a little more stability in your life, unless you love the adrenaline rush of working in the small companies. But that's just me, and you'll have to figure that out for yourselves, of course. And, well, yeah, what does work-life balance mean anyway? In a small company, you will never, you think you're working hard here, you just wait. Okay, so enough preaching. Here's what the main part of the talk is going to be. Biologics, it's all about biologics with an emphasis on monoclonal antibodies. So biologics place in the pharmaceutical market. High-level overview of the cost, time, and complexity of biologic drug development. Other speakers have covered some of this, so it'll be a high-level overview. Choosing a large versus small molecule for an indication, types of large molecules and what they can do. Identifying, optimizing, and developing a large molecule therapeutic, and how can this be done in the academic environment. So I'll go back 30 years when, I guess for many of you, that's when your parents were in diapers. But I was a few years into my career then. And it was really right around the middle to late 1980s is when the first, I wouldn't call it a wave, I would call it a ripple of biotechnology products came to the market. And in 1988, these are the 5 that were available. We have in the room one of the originators of Activase, Ted, over there. So what is it like now, 30 years on? So in 2016, 3 of the top 4 best-selling drugs were biologics. 7 or 8 of the top 10, depending on what source you find, and account that were over 60 billion in sales. And 7 of the top 12 bestsellers of all time, lifetime sales, are biologics. Now I'll point out, no biologics are in the group of most prescribed drugs. None, 0. Not even in the top 20, I don't think. So why do they make it into the best-selling drugs? Well, they're expensive. And believe me, for most of these, you don't want to have to take these because it means you're very sick with something. Anyway, they've earned their spot in in the pharmaceutical marketplace. Looking back over the last several years, anywhere from around a third to around a quarter of new drugs approved each year are biologics. This slide, until this year, for 3 years running, I didn't have to change it. This year, there's a new kid on the block, Prevnar 13. It's a pneumococcal vaccine. I think one of the reasons it's a bestseller is because they give it to aging baby boomers like me. I just took it. Fortunately, Medicare, I don't have to know how much it costs. So another way to plot this out is in this little colorful pie chart. Humira is the biggie. These are the 15 best-selling drugs. 10 of them were biologics. I think I went through and added it up. It was something on the order of 75 to 80 billion of these in sales of these 10. Of the 10 biologics, I think half of them were therapeutic antibodies. The rest were different things. This is, where is it? Prevnar is a vaccine. This is an FC fusion. This is a therapeutic, essentially a modified version of a cytokine. Lantus is a modified version of human insulin. But therapeutic antibodies currently are about half of this group. So as other speakers have told you, developing a new molecular entity drug is pretty tough, complex, challenging, and costly. It can take, optimistically, 10 to 15 years, about $1.5 to $2.5 billion, depending on how you calculate things and depending on how lucky you are and what kind of trials you have to do. But the time frame has an interesting implication to go from here all the way down to here, in that 15 years means that about a person's career in the industry spans slightly more than just 2 drug development cycles. All right? So for example, as I probably will point out later, forgetting that I mentioned it now, you know, if you and your partner have your first kid here, if the project goes well, by the time you get down here, they'll be in high school. Okay? It takes that long. Furthermore, as has been pointed out, there are high failure attrition rates, and it's not just getting to the approval and launch. Even after the launch, post-marketing surveillance on tens or hundreds of thousands of patients compared to thousands in the phase 3 trials can uncover safety-related issues that you didn't predict that will have a serious impact on how physicians want to use your drug and how many sales you get. So the risk doesn't stop once you've got it approved and set it up for sale. So how do you justify this monumental effort and associated risk? So there better be a significant medical need, as others have pointed out. You really need to understand the disease pretty well to be able to identify how we're going to interfere. You know, what molecules are we going to either stimulate or inhibit as a way of stopping or slowing down the disease process. And can we do that without unacceptable safety risks? So you don't want to make the patient worse, obviously. You want to make them better. So targets that have an impact on human disease are pretty rare. It's only about, I don't know, something like 500 out of the 25,000 known genes in the human genome have been identified as potential drug targets for marketed drugs. Is a candidate target critical to development or perpetuation of the disease? That's really important. Otherwise, you don't want to spend time and resources, and you won't benefit patients. So what type of therapeutic can modulate the target to ameliorate the disease? So a whole bunch of choices you hear a lot about in this room about small molecules. Therapeutic proteins we'll talk about today. There are also peptides, nucleic acids. I won't touch on that. Cells. You'll hear there are some projects in Spark that have involved cell therapy and gene therapy. So we'll now move those latter 2 points up to the top and say, and from the perspective of students and postdocs, what you generally focus on most of the time is identifying and validating targets as potential ways to interfere with or cure disease. So I pose this question because I would have answered this yes when I was a graduate student or postdoc. Identifying and validating new targets is the most challenging and intellectually demanding part of drug development, right? No, not necessarily. It's very important, but it's really only a tiny part of the drug development process. So we're kind of going to, before we dive into some details, we're kind of going to walk across this in broad brush for biologics. Right. When you discover a target, I'm presuming here now that you've identified and validated a target. So how are you going to interfere with that target or activate it? Well, would it be appropriate for a small or large molecule? How do you decide that? What do you want the large molecule to do? What kind of large molecule is the best way to address it? And what properties do you want to have? You start to think about and act on those questions in discovery. And then as you progress to screen, hit to lead, and lead optimization, you really focus on what properties you want it to have. Is the large molecule manufacturable, and can it be stably formulated for the desired dosing? This is one of the most expensive and resource-intensive parts of biologics development. It is more risky and complex by far for large molecules than it is for small molecules. And in fact, it not only occurs here, but it stretches out well into phase 2 and even into phase 3. Is it safe, and does it affect the disease or biomarkers in preclinical species? That's preclinical. You probably figured that out. Is it safe and efficacious in patients at doses that are clinically and economically feasible? That's what clinical trials are for. And will regulatory agencies approve it? Will it be safe, effective, and provide value for patients? So, particularly with regard to these last two questions, when you're pondering starting a project, a significant degree of clairvoyance 10 to 15 years into the future is important. It's not, of course, you can't, nobody's perfect, but you really have to try to imagine, look forward 10 to 15 years. Don't address what the problem is now. Imagine what the problem will be in 10 to 15 years. What other alternatives will there be for patients? And just to sum it up, begin with the end in mind as much as possible. So small or large molecule drugs. So these guys are small and these guys are large. You can see that, the molecular weights of these guys are in the thousands, tens of thousands, or hundreds of thousands. You probably know, everyone in the room probably recognizes this one. This one helped pay my salary when I was at DNAX Chewing Plow. It's loratadine. It was known at the time as Claritin. And this one I took whenever I contemplate giving a talk like this one. It's Zantac. Right? And so how do you decide? Well, what kind of target do you have? Where is it located? It both, in the body, but also in the cell that expresses it. What mode of action do you need? Do you need an agonist? Do you need an antagonist? Or do you need to kill cells? What is the disease, and what's the risk-benefit analysis? And what is the competition? Are there already a whole bunch of other people with active IP in that space? Are they years ahead of you? That's a factor in deciding to take on a project or not. And what modes of dosing are acceptable? That will depend, if it's a life-threatening disease, almost any mode of dosing will be acceptable. If it's just something that's a minor inconvenience, but a minor inconvenience to a lot of people, so a large market, you're going to have to be pretty demanding about the convenience and ease and lack of pain associated with the dosing. So large molecule advantages. So for large molecules, targets typically are secreted or cell surface protein targets. You can large molecules are generally pretty good at inhibiting protein-protein or receptor-ligand interactions that have large contact surfaces, certainly many too large for a small molecule to be effective. Agonist activity on complex receptors, such as cytokine receptors that have more than one receptor subunit. What are the drawbacks? Well, things like enzymes, whether inside or outside the cell, are generally better targets for small molecules. Cell surface molecules that are multi-spanners or receptors that have deep pockets for the ligand, ion channels and GPCRs, they can be challenging for large molecules. Again, probably a better bet to go after small molecules for those. Large molecules have very limited or no access to either intracellular targets or to the central nervous system unless you directly administer it intrathecally. Most, in contrast to small molecules where safety issues can come out of the blue or out of left field and slap you in the face, most safety issues around large molecules, most but not all, are related to the actual biology of the target and the mechanism of action. And where people can get caught up or surprised is when the biology of the target is not the same in humans as it is in tox species or in animal models. And that happens far more often than people think. Right? Dosing, well, you can do an infusion or you can go to the doctor's office and sit there and get something infused, IV. You can do your injection with a device, even do it yourself. The ability to dose can be limited by the viscosity of the formulation. These are big proteins, and if you concentrate them down, many of them become quite viscous, and boy, if you're going to give yourself a sub-Q injection with something that's viscous, you better have your pain meds at hand. And it may not penetrate tissues well, because it's large. Pharmacokinetics, so biologics tend to have pretty long half-lives, days to months. Particularly therapeutic antibodies can have very long half-lives, and sometimes the pharmacodynamic half-life, in other words, the duration of the biologic effect, can actually exceed by a lot the ability to detect the presence of the antibody in the system. Right? So good coverage of the target may result in better efficacy and less frequent dosing. However, there's a dark side. In fact, they have longer half-lives means they stick around a lot longer. So if there are safety issues, it's going to be hard to, you just have to wait it out sometimes, weeks, months. And large molecules can do things that small molecules cannot. Obviously, antibodies not only bind antigens, but they have effector functions. They can activate functions on cells. You can deliver toxins to particular cells that express the antigen that your antibody binds to. You can even, with therapeutic antibodies, and this is relatively recent, have agonist therapeutic antibodies for multimeric receptors. This was kind of a surprise to everyone, but a good surprise. And then cell-cell bridging, and we'll come back to that. There's at least one oncology therapeutic that brings cytotoxic, that brings T cells directly to the cancer cell and then activates to kill. So we'll decide on large since we're talking about biologics today. So what other types of protein therapeutics out there? Well, antibodies is what we're going to emphasize today. They're principally used as antagonists and target cell killers. I put a few examples here. Prolia and Xgeva are an Amgen product, interestingly. They're anti-rank ligand, I believe. And one is an oncology product for oncology. One is a product for postmenopausal osteoporosis. The doses are very, very different, and the formulations are different, so they're essentially separately branded. This one is is an example of an antibody drug whose pharmacodynamic half-life far exceeds what's known about the physical pharmacokinetics. The biologic effect persists many, many months longer than it takes for the antibody to disappear from the system. And in fact, this one, I believe, is only dosed twice a year. Yeah. The cancer one, of course, is dosed more often. Then there are recombinant proteins. These, in fact, were the first biologics used. They're mostly endogenous ligands or enzymes. It can be agonists or that stimulate processes, and there's at least one natural antagonist, IL-1RA. It's called Kineret. It's not used very much. It has terrible PK. And then there are Fc fusion proteins. One of the very best sellers on that little pie diagram I showed you, Enbrel, developed at Immunex, and then Amgen acquired Immunex. That's a TNF receptor Fc fusion, soluble TNF receptor Fc fusion, and it's a TNF antagonist. And there are a number of others. Eylea also made it onto the bestsellers list this past year. And then thrombopoietin, is this working? Okay. A thrombopoietin agonist mimetic peptide fusion called Nplate is peptides that bind to and activate the thrombopoietin receptor, and then this is antibody Fc to prolong PK and to provide cross-linking to make it divalent. This was developed rather than developing thrombopoietin because, as we'll find out in a few minutes, some native proteins are immunogenic, and if you're dosed with that native protein and develop a response to that, chances are it's going to cross-react with your own thrombopoietin and make you worse off than you were before. And in fact, there were some deaths with thrombopoietin, but not with Nplate, because it's not immunologically cross-reactive. So what do you want the large molecule to do, and what kind of large molecule? So agonist, antagonist, target cell killing. As we said before, endogenous ligands or mimetics as agonists generally. The ligands themselves are generally full agonists. Enzymes, of course, will do the job, the same job as the native enzyme. And the doses tend to be relatively low, sometimes just micrograms per kilogram. The disadvantages is that they can often be difficult to manufacture. They're often unstable. And many times you have to engineer them to convert partial agonist mimetics into full agonists, or engineering or post-production modification to enhance pharmacokinetics. Every time you modify it, you run a further, an additional risk of immunogenicity and via epitope spreading to the natural part of the molecule, immunological cross-reactivity. So immunogenicity may have significant associated risks, such as cross-reactive autoimmunity. So I just want to point out, for small molecules, safety is, risks and safety are as long as it's in your system. But autoimmunity is forever. If your therapeutic induces autoimmunity, you are stuck with it. And you have to live with the consequences or be medically treated for the consequences for the rest of your life. More recently, as I mentioned, agonist antibodies have made their way, at least to the clinic. I'm not sure about actually to the marketplace yet. So I think these are going to where they can be made will probably be the preferred modality. So Fc fusions, they're made from receptors usually, so they're specific and they're pretty high affinity. Many receptor ligand systems many receptors have multiple ligands, so if it's desirable, you can act on either multiple receptors or block multiple ligands. Sometimes that's a liability, sometimes it can be a benefit. PK often allow dosing every 1 or 2 weeks, so that's not bad. But they can be difficult to manufacture or unstable, and again, immunogenicity to the native part of the molecule has the risks that we just talked about. Antibodies are increasingly taking over for the last more than 15 years or so as antagonists, and as you all know, they're specific, they have high affinity, their PK half-lives are at least a week and sometimes as long as 3 or 4 weeks. As a class, they're really friendly to manufacture and formulate, although there are individual exceptions. The risk of immunogenicity is just infusion reactions or a loss of efficacy. So you don't generate autoimmunity to the target using therapeutics like this. The difficulties can be that species cross-reactivity can be limited, so it can be really hard to do tox studies if you don't cross-react to the tox species target. And they're large, so you need, for antagonists, you generally need pretty high doses, so you need 1 to 10 milligram per kilogram doses. For infusions, that's not too much of a challenge, but it can be a significant challenge if you want to give sub-Q or direct injection doses. And limited tissue penetration, it depends on the particular case. Target cell killing. So these are all the different ways. How many? There must be 30 ways to kill your target cell. That was a song that someone wrote, I think. Many of these are, at this point, just imaginary. They haven't been translated into products. The exceptions that I'm aware of are immunotoxin conjugates, and then this is an interesting one, a bispecific SCFV, a tiny little protein, which was approved in 2014. And they're both two little FV fragments, one specific for CD3 on the T cell, and another specific for CD19. So that's kind of the T cells depicted as a European-sized golf ball, and the leukemic cells depicted as a North American-sized golf ball here. But it bridges them and activates the T cell, and that contributes ultimately to killing of the tumor. It's a pretty effective drug. Lousy PK, but it's given by a device that you wear and continuous infusion. So smaller therapeutic protein scaffolds, there's still work going on. I said 4 years ago that their work's in progress. That's still true. There's one Kunitz domain therapeutic approved for hereditary angioedema, I believe, and that was approved back in 2009. There are a couple things in phase 3. There's one, Ablinks has filed a, what they call an antibody, a camelid antibody for, I'm blanking out with the anti-von Willebrand factor antibody. They have filed a BLA on that. I don't know the status of it. So there are, potential advantages, sort of the pitch deck that you would give, to investors if you were talking about it, which might make people experienced in the field kind of roll their eyes a little bit, but these are the things you would say. They're small, potentially better tissue penetration, favorable for both agonist and antagonist targets. They may access targets like, G-coupled protein receptors, ion channels, or enzymes. Can be challenging for antibodies. That's probably true. No glycosylation sites. You'd engineer them out if they were there, and some can be manufactured in E. coli. So the difficulties, as with all other alternative small format protein therapeutics to date, are 3. PK, generally the PK stinks, right? You have to at least make an FC fusion out of it. Manufacturability, things don't express very well. They're not very stable. And frequency of immunogenicity to the peptide or novel scaffold can be high. So that doesn't necessarily lead to cross-reactive autoimmunity, but it can mean that the more you have an immune response to your drug, it becomes ever less effective. Just cleared out of the system very quickly. So these things have not come along as fast as people thought they would. Antibodies are still kind of the gold standard for biologic therapies. Now, so since I mentioned PK, yeah. I'll come to that. Yeah. Protein pharmacokinetics is a little bit different. Werner, you can fact check me on these things, but protein PK also influences what kind of large molecule you're going to choose and how you're going to make it and how you're going to dose it and how often. And the basic thing is that kidney filtration of proteins, it depends on their size and is considerably less efficient for molecules with molecular weights greater than about 70 kilodaltons. So if you have a protein therapeutic that's in this range, is small, it's going to be cleared very rapidly through the kidneys. And then there's a gradual transition to, just based on size, it will be cleared much less rapidly if it's above 100 to 200K. So size matters. And a lot of approaches have been, particularly with native ligands and cytokines, is to make them look bigger to the system, to the organism, so they aren't efficiently filtered out. And you can make an FC fusion, like some of the ones I showed you. And it's nice because it's just a new protein that you produce. There's no post-production modifications. But fusion proteins don't always express well, and the activity can be lost with fusion, lost or greatly reduced. You can add a polymeric mass. Usually it's polyethylene glycol. So an example of that is GCSF, which was filgrastim. It's only got about 3.5-hour half-life, all right? But when you pegylate it, half-life goes up to 15 to 80 hours. Consequently, the dosing is much less. What I want to point out, although I won't show any examples, is that more often than not, when you pegylate, you lose efficacy. I mean, it doesn't go away, but it becomes a less potent molecule and sometimes a partial agonist. But very often, you will more than gain back through exposure what you've lost in efficacy. So you get increased PK, low immunogenicity risk, as was found empirically, and there are site-selective chemistries, so you can get a relatively homogeneous product. But as I mentioned, there can be activity lost, certainly additional expensive process steps, analytics, cost of goods, viscosity issues for pegylated proteins, and some toxicology things that you have to worry about. Well, it's not only size that matters, but albumin and some IgGs have much longer half-lives than you would predict just based on their size. And probably most people in the room know the reason for that. It's the FcRn receptor that recycles those classes of IgG and also albumin and instead of directing it to the degradation pathway when IgG and albumin and other serum proteins are taken up by pinocytosis. They go to the endosome where it's acidified, and then suddenly in acidic conditions, the FcRn receptor binds both albumin and IgG and prevents them from going to the degradative pathway and sends them back out to the outside of the cell. And when the pH returns to normal, they disengage and they're released. So how do we measure the pharmacokinetics of therapeutic antibodies? So it's actually a very interesting problem when you think about it. The problem is to measure and detect small amounts of therapeutic antibodies, shown here in red, in an absolute ocean of nearly identical endogenous human immunoglobulins circulating in serum. So what distinguishes therapeutic antibody from other circulating immunoglobulins? It's variable regions and antigen binding site, which is called the idiotype. So those are up here. This is an antibody molecule, FC, in the variable regions. And the idiotypes or idiotopes are generally involved the complementarity determining regions, but not just those. You can make anti-idiotype antibodies that do not interfere with the binding of antigen to the antibody. But in any case, an antibody against your therapeutic antibody that specifically recognizes this region will be blind to the rest of similar class immunoglobulins in the serum, and that's a good thing. So these are examples of typical PK assays for therapeutic antibodies. One would be target capture, and the other would be the anti-idiotype. Let me back up here. So you can coat with the recombinant antigen, recombinant target, or you can coat with the anti-idiotype antibody. And then you put your serum or plasma gamish on there. It contains your therapeutic, which will bind, and a whole bunch of other irrelevant stuff. And then you wash away the irrelevant stuff, and you come in with a detection antibody conjugate. Now, one thing I'll point out here is that a potential difference in the assays is that the target capture assay will not detect antibodies in plasma or serum that are already bound to the antigen. Okay? They won't bind there. Whereas this one, if you choose an anti-idiotype antibody that doesn't interfere with antibody antigen binding, then you can detect those. What you'll notice is that all of a sudden you've had to do a second antibody project. Not just your therapeutic antibody, you have to make an anti-idiotype antibody. And that can sometimes be considerably harder than it was to make the therapeutic antibody in the first place. So some other things that influence PK of proteins. I'll just go through them. Proteolysis followed by renal clearance. That's sort of the analog of small molecules, getting broken down. The difference is that we end up with peptides and proteins and amino acids that generally have no biological activity, whereas small molecule drug breakdown products can do nasty things that you never guessed they would be able to do. Target-mediated clearance. If there's a lot of your target, it's going to soak up what you dose. And you'll have to give a lot of it in order to see the therapeutic effect. So it can be, if cells are in the subcutaneous space and you give a subcutaneous dose, it's going to get, a lot of it's going to just sit down right there and not go anywhere. If the target is expressed on other abundant short-life cells, red blood cells, platelets, neutrophils, they'll bind to those cells, and then those cells get cleared out pretty quickly. And target-mediated clearance, the target itself, particularly if it's a receptor, can clear, can reduce the half-life of your therapeutic antibody by receptor-mediated endocytosis and catabolism. And then what a lot of people don't think about is that an abundant soluble target that's produced at a high rate in the body can overwhelm the drug, and you just can't give enough of the biologic to really effectively inhibit it. So what properties do you want the biologic to have? So now we're in the screen and hit to lead area. So how do you get these things? Well, for recombinant proteins and Fc fusions, usually you already have the cDNA for the cytokine or the receptor, and you clone and express it as a fusion protein and potentially engineer it to optimize its properties as a therapeutic. Some of that engineering is empirical, things to increase PK or increase stability. For antibodies, there's a number of different ways. You can humanize a rodent antibody, which is probably what, for any project that's done here, that's probably the way it will be done. You can use phage or other display technologies, or you can use humanized animals, usually humanized mice. But this is pretty specialized. Those mouse strains tend to be pretty wimpy in terms of their immune responses, and it takes a facility and people with a lot of experience coaxing efficient fusion and efficient immunization and efficient antibody production out of those animals. So I would not recommend that a place like Stanford just bring in, even if it were available, that you just bring in this mouse strain, because you're going to be very frustrated, very disappointed. You'd better also hire someone who's worked with it and really knows what they're doing. And other scaffolds that I mentioned, like I said, they're still works in progress, haven't made a big impact in the field, but it's generally just display technologies. So what did we get from this screen? Whether it's screening monoclonals or phage display or whatever. Well, is it specific for the target? That kind of seems obvious, but does it bind to the target? Does it bind, importantly, does it bind to the tox species version of the target? It's not impossible, as Mike Taylor, I think he's here today, will tell you, it's not impossible to develop a therapeutic antibody that does not cross-react with the generally the cynomolgus monkey ortholog. But it's really difficult, and it's slow, and it can be expensive, and companies that may think about licensing your antibody may think a little bit about, do we want to support that extended timeline and that extra cost? Does a large molecule modulate the target as desired? Does it modulate the tox species target as desired? You want to know the on and off rates, the affinity for the target, how potent is it, not just its affinity, but how potent is it in a bioassay of biologic activity of the target? Whether it's an agonist, you get an EC50, or an antagonist, you get an IC50. And among the antibodies that you get, how diverse, how many different epitopes do these antibodies bind to? And that's important not only for a potential pivot during the development process, but it's important for IP coverage. You want to tie up as much of the target molecule as possible to keep others from playing in your sandbox. it's very important for those of you who may be working on this type of thing to have all of this thought out and appropriate assays in place. Because if you don't, like anything else, if you don't know what you're looking for, you won't get it, right? And if you can't assay what you're looking for, you won't know when you've been successful, and you probably won't be successful anyway. So it's really important to think through the project before you start immunizing animals, before you start screening antibodies. Okay? So what properties do you want it to have? Is a large molecule manufacturable? Can it be stably formulated for desired dosing? So lead optimization in small molecule world is generally medchem, right? Here, it's mostly looking at how you're going to manufacture and formulate it. And it's really expensive to do that. So this is just, a diagram of antibodies. I won't go into this. You probably already know it. But one thing I wanted to point out is that there are all mouse antibodies, and they end in the suffix OMAB. There are completely human therapeutic antibodies. They end in UMAB. And then chimeric antibodies where all the constant regions have been made human, but the variable regions are completely mouse, end in eximab, and an example is Rituxan or rituximab, okay? And then humanized antibodies have this suffix, so you can tell from sort of the antibody name, not the trademark name, but the antibody name that's given to it, which type of therapeutic antibody it is. Well, one of the things you want to ask is, all right, we have our therapeutic antibodies, it meets all the requirements 2 slides ago. What ice type should we use? We could, if it's an IgG 2 or 3, we can change it if we want. Well, how would you know if you want to change it? Well, do you want effector function? For the biology that you're going to generate with your antibody, do you want effector function or not? Okay, maybe you do, maybe you don't. That you have to know the biology of your target. So IgG1 is really good for that, but there is a potential for light chain reduction in the manufacturing process. The next choice, if you don't want effector function, is generally IgG4. I think, what is it, the CD90, which antibody was Herb Weissman's group developed? CD90? CD47, okay. I think that one was an IgG4, all right? They have low effector function, which is good, but heavy chain mixing occurs when you inject an IgG4 therapeutic antibody. If you wait long enough, it starts out dimeric, but it'll eventually swap with other random IgG4s, and you'll end up monomeric. Heterogeneous monomeric population that is only one of the chains of the IgG4s that you would find bind the target, the other some random thing. Okay. IgG2 and IgG3 aren't used, particularly 3 isn't used all that often. This has the least effector function, but it has some manufacturing issues. Generally, the preference these days is that if you want to eliminate or minimize effector function, you'll do some mutations of this one in the Fc domain rather than go to 2 or 4. So, getting from a large molecule lead to a large molecule drug, we talked about choosing the heavy chain, but you may find that when you sequence the variable regions that there are some things that maybe you wouldn't be too happy about. Extra glycosylation sites right in the middle of the variable region. When you manufacture it, it's not all going to be one, it's going to be heterogeneous. All glycosylation is heterogeneous, no matter what cell you use. There could be unpaired cysteines. That's going to make it a nightmare as far as aggregates are concerned. Potential oxidation, deamidation, isomerization sites are all going to contribute to the heterogeneity of your product and potentially irreproducibility from one batch to another. And of course, potential proteolytic sites, which is not a good thing. If your favorite antibody has one or more of these things, you can try to engineer them out. As often as not, that greatly reduces or even kills the activity, and you may have to just go to another candidate antibody. Glycosylation, not usually all that important, but for antibodies it has some potential importance in that if you want effector function, both antibody-dependent cellular cytotoxicity and complement fixation are highly dependent on glycosylation of the antibody. In another example, erythropoietin has 4 glycosylation sites that seem to be important for stability of the protein. If you get rid of them, it's a much nastier protein to work with. And not only function, but potentially pharmacokinetics. So if the host cell that you're using to produce the protein puts predominantly terminal galactose and mannose on the carbohydrates, then protein's going to be cleared much more rapidly by scavenger receptors. In contrast, if you have terminal sialic acid, they're not cleared as rapidly by these receptors, and it also minimizes the interactions of the extracellular matrix and resulting and associated kidney clearance. So Aranesp, which is an engineered version of EVO, has 3-fold improved half-life because 2 more glycosylation sites were added, and it's most likely because of the additional negative charge. Whoops. Okay. What am I doing here? All right. And then as we discussed, you can engineer, instead of choosing another isotype, you can, if you want to just stick with IgG1, there are mutations now that are well characterized. Probably have to license them because they're patented, where you can reduce binding to Fc receptors, you can reduce complement fixation, and you can even augment the FcRn-mediated binding and resulting half-life as well. Okay, not all antibodies are good drugs. Some of them aren't expressed very well, believe it or not. When they're expressed and processed, many of them can aggregate and become very viscous. Signal peptide can be cleaved heterogeneously or not very efficiently. There can be glycation, oxidation, isomerization, and the formulation has to be stable to temperature, light, to pH changes, because whatever you formulate it in, when you inject it, it's going to be pH 7.4 or awful fast. And so here's an example of what's called a pH jump assay, where an antibody, this was done at Amgen as an example, was added. The formulated antibody was formulated at, I think their buffer is around 5, 5.5. Added to water, it stays in solution. You add it in progressively greater amounts to essentially a solution that buffers the pH at 7.4, and you've got precipitate. Okay, not a good thing. It'll reduce the effectiveness of your drug, and these tend to be pretty immunogenic, these precipitates. Is it manufacturable? Can it be stably formulated for the desired dosing? So generally, therapeutic antibodies certainly are produced in mammalian cells in various strains of CHO cells. Some other types of therapeutic proteins can be produced in E. coli. There are relatively few. Some vaccine proteins are produced in yeast, but yeast has not been very popular. So you need to evaluate expression levels. You need to develop a purification process and see what the yield is, and the characteristics, the biochemical characterization, which I'll show you in a minute, some examples of the product. And then when you're satisfied with all of this, you select the final molecule, you validate the DNA and protein sequences, you have your target product quality profile. That means what are the analytics we're going to use to compare batch to batch, and how much difference can we tolerate, can we in the FDA tolerate in those characteristics. You generate your cell line, and then final cell line. This is generally where good manufacturing process begins, or GMP. Well, then you have to make GMP cell line, cell bank. So all under GMP, where documentation is the word. You make a culture of the recombinant cell line. You aliquot it into a relatively small number of vials, 60 or so. That's called your pre-master cell bank. Each one of those can be expanded to about 300 vials into a GMP master cell bank. And then each one of those, each time you do a production run, you can make your GMP working cell bank and use one vial to inoculate a culture for a production run. So you can see the power of compounding here. Generally, by the time you've gotten to this stage, you have many, many vials. You're able to do, if necessary, 250,000 production runs that will last about 1,000 years, and that's way beyond even the patent protection that Stanford's IP people can give to you. So these are some examples of, by the time you are going to be producing kilograms or even tons of therapeutic protein, you may be growing mammalian cells in 15,000 liter, multiple 15,000 liter bioreactors. These are chromatography columns, believe it or not. I've seen these. They're, several feet across and a couple feet deep. I believe the ones I saw were protein A. One of the biggest support functions for the biotech industry is the production of protein A to make protein A resin to purify therapeutic antibodies. And then these are just, I think, the sort of bottom floor of some of the bioreactors. So in the old days, meaning 10 or more years ago, it used to be when you had a therapeutic antibody candidate, you had to build a whole new manufacturing facility just for that therapeutic antibody, which was about a $400 to $500 million investment, and 4 or 5 years to get that done. So you really had to take the chance when you're sometime in late phase 2 or early phase 3 of saying, all right, we're pretty convinced it's going to work. We're going to shell out the half $billion to build the facility, and God help us if we're wrong. All right? So there's been a lot of work to try to make this process simpler and ability to do many different therapeutic proteins in one facility, miniaturize it. And there's been some success. I'm not going to go through all this. This picture's a little out of focus. I think it's allegorically apropos because the impact of these efforts is still not really in focus as to how impactful it's going to be. But it's very important. This is a huge cost driver in the development of therapeutic proteins. Okay, so what do you do when you run a batch? What have you made? Is it the same? Is it tolerably similar within specs to the last batch you made? Does it meet the release criteria or product specifications? So for a small molecule, comparatively speaking, that's pretty simple. For a large molecule, this is what you're dealing with. Okay? There can be aggregates. There's certainly heterogeneity of glycosylation. There can be some disulfide scrambling sometimes. There can be charge variance. So the point is not to throw up your hands about this, but the point is that you have to develop a process wherein the result is reasonably similar every time, all right? And develop, working with the FDA, with the regulators, you develop, just as for small molecule, product specifications and release criteria. But they're rather more complex than for a small molecule. And of course, then there's, is it the right molecule? Is it potent? And what are the other impurities? What other proteins? Proteins from the culture media or from the cells themselves? Is there DNA contamination? And any of the process reagents, sometimes protein A will leach off the column and you have to either get rid of it or define your specifications to tolerate a certain amount of it there. So the first cycle of purification gets you to what's called drug substance. That's where your drug is formulated in something like a 5-gallon container. You've expressed the product, you've purified it, maybe kilograms of the product. You've formulated it and you want to store it here, typically at 4 degrees, refrigerated at least for a number of days. And you have process and analytical controls around specifications for this. Well, then to get it out to the patients, you've got to take this and aliquot it into vials or injection devices. So that's the primary container. And you need a formulation, maybe a different formulation for that. You need to precisely regulate the fill volume, and you need to have the right, agreed upon packaging and labeling. Now, formulation is actually a pretty serious exercise. Kevin, I know you mentioned in one of your talks that, you know, one of the, I forget the context, but one of the formulation people you had asked had said, oh, it'll take, you know, many grams of protein in 2 years or a year and a half to work out. That was probably a person who does commercial formulation development, right? But typically there's first-in-human formulation development as well, and that is essentially just to get it out there into the clinic to see whether it works, okay? So this starts right away when you've committed to send something, you know, to develop something, and typically gets you at least through phase 1 and 1A and 1B and maybe partway through phase 2. And then once you're convinced it's safe and is doing something, you'll take the risk and start commercial formulation development. And that'll take you through phase 3. That's a much more serious and challenging effort because you ultimately have to have something that patients and physicians will use, and that's what you want to use in phase 3 trials as well. So you may have to have a very high concentration that's not very viscous. Not all antibodies will do that. You don't want particles. You don't want it to be green, like Steve was saying, something turned out in his small molecule projects, or you don't want it to be yellow like I saw with one of the avomers that I worked with at Avidia. It can't be, it has to be minimally viscous, appropriate for how you're going to get it into patients. Really, ideally, you'd like the drug in the container to be stable for 2 years at room temperature. That's a pretty tall order, but it can be done. It has to withstand it, especially here, multiple freeze thaws without forming precipitates or aggregates, or, and it has to withstand shipping and handling. So I just wanted to, I put this thing here because, and I mentioned this before, but one of the most, when I was at Amgen, they had a program where scientists in different parts of the development pathway would come talk to the discovery research folks like us to teach us what was really involved in the downstream stuff, and hopefully we would be able to do our part of it more intelligently to make their jobs easier, right? And it, I mean, it sounds cynical to say that, but it's not. It's really important because their jobs are far more time critical and expensive than ours. One of the speakers, she described a particular problem they wrestled with, in which how it ultimately turned out, and it was one of the most outstanding pieces of scientific detective work that I have ever seen, even though it wasn't discovery research. They found out that the vial they were using during freeze thaw would delaminate at the microscopic level. And it wasn't anything you could see visually, but it would result in particles of what was coating the inside of the vials being in the solution. Now, this never got into people. It was something they discovered during the process development. And it took them a long time and a lot of effort and a lot of very elegant analytical and detective work to figure that out. Okay, so you have to be able to do it reproducibly at scale. So like I said, first in human process development and then commercialization process development. I won't go through this because you'll all have access to the slides anyway. And again, in contrast to small molecules, a typical biologic manufacturing process is really complex. You have to have controls and GMP controls, GMP validation for each step, all the way from your cell bank vial into your seed culture vessels, into your production vessel, and then separating the cells out once your product is in the supernatant, going all the way through here, inactivate potential viruses, more virus filtration, more purification, then your drug substance in the 5-gallon or whatever container, more formulation, sterile filtration, final container, frozen liquid or lyophilization, and that's your drug product to send to patients. This is way different from small molecule drugs, and it's much more expensive. So is it safe? Does it modulate disease? So we're coming down to the home stretch here, and this is where things are not that hugely different from developing small molecule drugs. So, but there is one difference, and that is that for biologics, usually small molecules, you do 2 different species, sometimes not even a primate. Sometimes you do just a rodent and a dog. Here, usually, you're just using one species, non-human primates, and that is because you have, or ideally have, your therapeutic cross-reacts with the target in non-human primates. Remember, the main safety risk for biologics is the biology of the interaction itself, not degradation products. Okay? So there's, just like for small molecules, there's short-term toxicology, 1 to 3 months, chronic tox, 6 months. As you get towards phase 3, where you think it's really going to work, you do reproductive toxicology. Primates, that's, as you can imagine, a long-term study and very expensive, $1 or $2 million at least for a study like that. If the mechanism of action of your drug suggests a risk of inducing cell proliferation, then you'll have to do an oncogenicity study as well. And then safety and efficacy in humans is very similar. Single and multiple for phase 1, it's primarily safety, but including immunogenicity, pharmacokinetics, and you'd like to see if it's at least inducing the biology that it's supposed to do. So you would have biomarkers even in phase 1 studies. Phase 2 and phase 3 are very similar to the small molecule world. Now, I want to come back to immunogenicity, or the generation of anti-drug antibodies. So, you know, I'm not going to go through each of these. If you want to look at the slides later, they're, like I said, they're available. Sun has them. But I want to highlight what are the potential outcomes if your drug is immunogenic. Maybe nothing. May not matter. May not affect PK, may not affect activity or drug. It may, on the other hand, neutralize your drug's activity or alter its activity. Not a good thing. It may alter its PK, generally clear it faster than you want it to be cleared, although there are some exceptions. And again, I'll highlight this because it's really important, if not for therapeutic antibodies, but for other types of drugs where a native protein is part of the drug. You really want to be careful of this because it can be a really serious lifelong safety issue. Okay, so the important takeaway, I don't expect you to remember all that stuff, but the important takeaway that even a lot of people, many people in the industry don't fully appreciate. Immunogenicity, when you do your tox studies and you look for immunogenicity and you assay, well, is it developing an immune response? What's the titer? Is it neutralizing? Da-da-da-da-da. The important thing to remember is that immunogenicity in tox species, such as non-human primates, is not predictive of immunogenicity in the clinic. It isn't. But yeah, the immunogenicity in tox species, particularly neutralizing immunogenicity, it can make it difficult or nearly impossible to do preclinical safety. You know, if the immune response in the animals develops that neutralizes the activity of the drug isn't doing anything. It's like a protein nutritional supplement that you inject, right? So that's, when you're doing your safety and tox studies, that's the key issue there regarding immunogenicity. And will regulatory agencies approve it, and will it be safe and effective? So just like for small molecules, you file an IND when you've completed the appropriate safety studies and manufacturing documentation. And then once you've done all your clinical trials and you think it's worth moving it forward as a drug, you would do what's called a BLA, or biologics license application. And then you need, of course, regulatory approval to put it on sale and distribution in the U.S. and approval from the other nationalities or the European Union agencies as well. So kind of to summarize, so I'm not going to read all through this, but this just sort of shows you, this is summary A, covers from here to here all the various steps. What typically you guys will be doing or are doing is highlighted in the orange box. And you can see here, this little tiny box is target discovery and validation. Okay? So obviously it comes first and it's important, and there's no point in doing the rest of this if you haven't done this right. But this is a tiny part of the overall picture. Okay? Furthermore, if you're, and Kevin, you asked this question, I remember a year ago, he said, what would I recommend for people here developing antibodies? I would recommend that you essentially focus on this top line. And I wouldn't, you know, in the slide on humanization, I really would hold off on humanizing until you have something that you're really convinced has what it takes to be a therapeutic antibody, then send it for humanization. And then molecule to product, there's a whole bunch of stuff that we've talked about as well, and that covers this part. And now when that bouncing little baby is now a sophomore in high school, you may get your first product launch. And if you're lucky during your career, you may get to see 2 of these from beginning to end. So, how would one do this in the academic setting? So, obviously, financial and time considerations are limiting. So you need to really plan up front for success, right? Understand your target. This is really important. Garbage in, garbage out, folks. If you don't understand your target and its biology, and you don't know what the properties that you want in the therapeutic, chances are very low that you'll get something you can use. So you need quantitative assays for activity and for that of the proposed biologics, such as binding and function. Preferably assays that are relevant to and predictive of its in vivo activity. If possible, it may not be possible. You really want to have in hand the cloned, expressed, tox species target homologue, the cyno version of the human target that you're trying to make antibodies against. And ideally, your candidate therapeutic should cross-react to have an IC50 or EC50 within a factor of 10 of the human, of what it is on the human target. That will make development much easier. That will make it much more attractive to companies that think about licensing it. You want to understand the desired in vivo function of the therapeutic. So what measurable properties must it have? Have you identified a PD marker? How will you know it's working in vivo? Do you need effector function or not? Probably important to know. For an oncology drug, you might want effector function. For other things, maybe you wouldn't. And this will be part of target product profiles, as I guess some of you are going to start showing us in the next week. If any of them are for a biologic, you probably should have this kind of information in there. Ideally, what amount do you want to dose, and how do you want to dose it, and how often? Importantly, design your immunogen that you use for generating monoclonal antibodies with the above things in mind. Okay? You may not know the answers to all these things, but you really should put in as much effort as possible to get the answer to some of them, because that will inform how you immunize and how you screen. And then finally, as I mentioned before, don't humanize until you have at least a reasonable candidate according to the above. And understand that if you take too many shortcuts, if you ignore all of this, your chances of getting a developable therapeutic antibody will just be purely luck. Could happen, but it'll be just luck if you get it. Okay, for the final slide, I want to come back to the postdocs and the students. So, as you progress from here, maybe some of you are going to stay in academics, and that's great, and the supplies as well. Maybe some of you will go on industry, either large companies or small companies or both, but my 30 plus years of experience I've kind of distilled down is take these things that I think are key to individual success. Be excellent at what you do. And the industry has been either static or in decline for nearly 20 years. Unfortunately, what this means is that when you take a position, if you want to advance, average isn't good enough. You've got to be excellent. You've really got to stand out. So be excellent at what you do. Be ethical. First of all, it's the right thing to do. Secondly, it's a highly regulated industry. If you're not ethical, it'll catch up to you. And thirdly, what you're doing affects people's health and lives. I can't think of a better 3 reasons to be ethical. You should have the highest ethical standards, and there may be circumstances where you perceive it to your personal disadvantage or discredit to be ethical. Don't buy it. It's always better in the long run to ignore those feelings and do the right thing. Communicate, communicate, communicate. Make your collaborators feel like collaborators. Make them feel valued. You will do a better job in your work, and they will do a better job in their work, and that will inure to your own benefit. Play well with others. I can't emphasize that enough. Don't be an SOB. Treat people well, with respect, and they will respond. Seek out good mentors. If I look back over my own career, this is one of the things I did not do as well as I should have, and I think I would have benefited a great deal. But look for people in your company or elsewhere in the industry whom you admire and respect, and seek them out. They're only too happy to help. You know, after my own experience, I really enjoy mentoring younger people. It's one of the things that I don't get to do all that often, but I really enjoy it. Be aware of what's happening in the industry. I think that goes without saying. Just like in academics, you have to know the literature, right? Keep up with literature. Think like an owner. When you're, start out as a scientist or a senior scientist, think about how, what's the big picture of the company and how your work fits into it. And that will inform how you make your own priorities and how you do your own work, and it will only benefit yourself, your colleagues, the progress of your work, and the progress of your career. And then I'll close with that. So, we still have half an hour. Okay, thank you. Any questions? Some, but not all of the data. A lot of the production specs are available on, through the agency, but some are kept private. But importantly, one of the first steps in developing a biosimilar is to go buy the product that you're trying to copy and do the analytics yourself. Okay? There are still a lot of targets that it's difficult to make antibodies against them. I highlighted some things like ion channels and GPCRs. I think, if some either academic group or platform company came up with a way to really make that much easier and much more reliable, that would have a huge impact. I think a way to make, to expand the availability and capability of generating fully human antibodies. I think that would be useful. It's pretty specialized, even though, most people outside the industry may not appreciate that, but it is pretty specialized. It's not easy. Even if you have the animals, it's not easy. And then if there were a way that your panel of antibodies that you get when you screen a target were more regularly friendly as far as manufacturing and formulation, I think that would have a huge impact as well. So those are that's my personal opinion. I'm not familiar with the platform space myself, really. I haven't kept up with that. Yeah, just lay in a supply of Prozac. The high dose. Yes, but the issues are the same as with small molecule stuff, right? It's the CRO. Your project is never their job one, and they may not put their A team on it. Particularly if you're if you're a virtual company, you know, that's really just one or two people and trying to do everything through CRO, they may not put their A team on it. They'll reserve their A team for the larger companies that come to them and want to spend more money and have a potential for future projects. So it's a risk, but it's possible. You'll probably yell at your wife a lot, you know, when you come home from work every day. So.
For small molecules, yeah. Build a team of 5
So, yeah. Yeah. So let me relay some of my experience at Amgen, and then I've, you know, kept in touch with the people who stayed there after I retired. There was an effort, and this was really at the kind of discovery research and reagent generation level. But some of that was anti-idiotype antibodies to be used in development. Starting about a year before I left, was part of the reason I decided to leave, there was a strong push to outsource as many functions as we could to a CRO in India that Amgen had signed a long-term contract with. And it was tough. You know, one of the groups I was looking after was the research monoclonal antibody groups. And I'll have to say, and this is true before I came to the group, you know, I was just kind of the grandfather figure and patted them on the back and made sure that the groups at Seattle and South San Francisco and Thousand Oaks talked more to each other and coordinated their work, benefited from expertise. They were outstanding. They were the best group of their type I've ever seen. But the push was to outsource it to an antibody group at this company in India. And it was very frustrating. You know, I even went through the exercise of gathering all the data to show how much it cost our group to do a completed project. Because of our efficiency, it was actually cost competitive. And certainly it was more efficient. We could do more projects. It was a challenge, and it's still not going well 4 years later. So, and then there was protein expression, recombinant protein cloning and expression. Those things have not gone particularly well. There may be other success stories elsewhere in the industry, but according to the folks I know who are still there, it's been a real headache. And, you know, that's because they're doing the tough things. You know, if you want to just express a protein, there's already, you know, 15 years of literature on it. Well, that's one thing. But if you're looking at new targets and trying to discover and validate new targets, you have no idea how easy or difficult expressing, you know, an immunogen form of the protein that you're trying to interrogate. You don't know how that's going to be, what purification methods you're going to have to use, what analytics you're going to have to use. All of this is like, you know, people who've worked in the field for, you know, 10 years or 20 years, they know this shit. And their experience really makes it go much faster, much better, and it's right there. So anyway, as you can gather, I'm not a big fan of outsourcing and CROs, but I recognize their place in the industry, and I'm also glad I don't have to deal with them anymore because I'm retired. Yeah, I'm going to have to plead ignorance there. I'm not really all that familiar with it, so I can't help you there. Okay. All right, anyway, slides are available as a PDF, and if there are, you know, any in the group, if there are any projects for which you guys think a biologic would be an appropriate approach, you have my phone number. I'd love to help. Yeah, my wife will be grateful. Yeah. So. Yeah.